Our final talk of the evening is from Coralie Vacher and Cerissa French from Illumina. Illumina have been long-term supporters of this meeting, and I'm very grateful for their sponsorship of the continued sponsorship of the meeting. With that, I'll hand over to Cora to start off. Or yeah. Okay, great. Thank you very much.
And thank you very much for the PTC for giving us the opportunity of giving a talk tonight. As Sharon mentioned, I'm Coralie Vacher. I'm working in the marketing team at Illumina, focusing on application for complex disease, and from my training with a particular interest in neuroscience and brain disorder. I will cover the first part of the 25-minute talks, and then I will hand over to my colleague Cerissa. I will cover the sequencing side, and then Cerissa will have a presentation on our RA side with some very exciting new release that we're going to have later this year. Okay. So earlier this year at JP Morgan, we've introduced the new NextSeq 2000, and as you can see on that slide, it fits in our portfolio of sequencing instruments. So on one end, you have the more benchtop side of the instrument with the iSeq and the MySeq, and on the other end of the spectrum, the very much workhorse production scale instrument like the NovaSeq. And to give you an idea, with the MySeq, you get a maximum output of about 15 gigabase. And with the NovaSeq, if you use both flow cell and a maximum output, you get about 6,000 gigabase. So in the middle, the NextSeq are very much mid to higher throughput type of instrument. The new NextSeq 2000 provide about, or will provide, when the new P3 flow cell is released toward the end of the year, about 300 gigabase in maximum output. At the moment, it's providing 120 gigabase, which is the same than the NextSeq 550. The big advantage of the NextSeq 550 that we still have on the market is that it's got capabilities for reading our arrays. It's got scanning capabilities, and Cerissa will be able to tell a bit more about those later in the presentation. So we went through a complete redesign from the NextSeq 2000. By that, I mean, I'm not going to go through all the innovation that we went through because there were a lot, but we changed the chemistry, for example, from for a blue 2-channel sequencing by synthesis chemistry. We also changed the optics for much higher resolution, which makes the read and the run a lot faster. The flow cell also have been changed. They are much denser with a pattern. They're also a lot smaller compared to the NextSeq 550. Last but not least, we have integrated our Dragon hardware in the instrument, and that's the first instrument that comes with the Dragon hardware, and included a number of bioinformatic workflow and pipeline in the instrument. So this really allows the instrument to give power for much higher throughput application. I say that compared to, for example, the NextSeq 550. And we did that for 2 reasons: to allow our researcher to expand their project in number of samples to gain power, or to scale up in terms of depth of study. So allow very much multi-omic. And I would say that's really much the instrument that we have released with multi-omic in mind. It's also a lot more simple than the NextSeq 550 in terms of bioinformatic analysis. It's a lot more push button, and it's also a lot faster. So to give you an idea on the spec, at the moment, the instrument is shipping. It's been released with the P2 flow cell. and toward the end of the year, if COVID doesn't delay things, we will release the P3 flow cell. Both flow cell will give the quality that you can expect from Illumina, so over 75% of Fred 30 data. At the moment, as I said, the maximum output for the instrument is 120 gigabase, but with the P3, you'll have 300 gigabase. In terms of time for run, it takes just a little bit over a day. And I think for a lot of people, the point is to try and compare how many sample they could do, or many gigabase they could do for their with their budget. With the current P2 flow cell, it costs list price, about $30 per gigabase. And toward the end of the year, when we release the P3, it'll be more towards $20 list price per gigabase. To give you an idea, for the NovaSeq and the smallest flow cell, so the S prime, the list price is at around $15 per gigabase. So for me, that's specs, and usually that doesn't talk at all to me. So we included a slide that gives an idea more in terms of what does it mean in terms of experiment. At the moment with the P2 flow cell, for all exome sequencing, you can do about 16 samples if you want to do 50-fold coverage, or toward the end of the year, about 40. And it's quite linear. So I know most people would go more towards 100 mean target coverage rather than 50. So it's about 8 samples and 20 samples. We do have a lot of people doing single cell at the moment, and I saw quite a few presentations, in particular Julian's presentation. And on the current P2 flow cell, you can do, for example, about 10 samples, for which you would select 4,000 cells per sample and try to do 10,000 read per sample. So that gives you a good quality for gene expression profiling, if you want a comparison. Oh. My slide are not yet changing. As I mentioned, so we have included the Dragon hardware in the instrument. I don't know if everybody is familiar with Dragon. In February, we announced that we collaborated with the Broad Institute to develop a Dragon GATK package, which increased the accuracy and makes the alignment and calling a lot faster. If you're interested, there is a very nice blog on the Broad website about that. And so we integrated the Dragon hardware in the instrument, and then you can use the instrument either on site if you'd rather use your own server, or if you're not allowed to use the cloud to analyze your sample. And it also works with base-space sequencing hub, so on the cloud. And if you require to, or if you have the capabilities to use your on-site system, but you do need more capacities from time to time, you can switch from using your server to the cloud and back to your server. And at launch, we're releasing a number of bioinformatic pipeline around enrichment, RNA analysis, and germline analysis. And the instrument generates FASTQ, so if you want to start your analysis from scratch from FASTQ data, you can absolutely do that. In terms of library prep, so we are releasing a number of library prep this year. I'm not going to go through a lot of detail, but just in general, we do have 3 key library prep technology. Library prep that are for genomic DNA that are now used using bit-link transposon, and I'll go on that in a minute. Our TrueSeq ligation library prep around RNA-seq analysis. And of course, for people who are interested in more targeted sequencing application, whether it's for RNA or DNA, we have the AmpliSeq for Illumina portfolio. So the Nextera DNA Flex whole genome sequencing, or PCR-free library prep, is very much an innovation that we've released a couple of years ago. And basically, you can start from purified genomic DNA, but you can also start from raw sample as an input. By that, I mean either blood or saliva, or at the moment, things more like microbial culture or microbial sample. And the library is linked to a bit that has the primer and the transposon, and you go from your raw sample into a library that's ready for sequencing in about 3.5 hours. So that's very much increasing the speed of the workflow, and it also enables to really streamline your protocol and have way less hands-on time. It can be automated. It has the usual high performance you can expect from Illumina, and as I said, it is you can use it directly from raw sample, and you do not need to do qPCR quantification. So it really help with the COGS and the labor in the lab. It's also suitable for high throughput with 384 dual indexes. So that's for genomic DNA. I'm struggling to change my slide. Sorry. Okay. And in terms of RNA solution, we do have a number of new RNA solutions for messenger RNA or for total RNA for transcriptome analysis, with a new ribodepletion solution. And again, for people who are more interested in targeted RNA analysis, we do have a solution for enrichment. So on the new NextSeq 2000, for example, you would be able to do about 40 samples for a basic differential expression profiling, where you don't need a lot of reads for each sample. But equally, because of the capability of the instrument and the fact you can go for 300 gigabase of higher throughput, if you require to do things that are more greedy in terms of read per sample, like alternative splicing or a de novo assembly or allele specific expression project, you can really do those on that instrument. And again, this is entirely supported by the Dragon pipeline. And I'm going to hand over to Cerissa, who's going to cover our release around microarrays.
Thank you, Cora, and thanks everyone for staying till the end of this session. It's quite late in Europe, so I completely appreciate it. So as you can see from Cora's presentation, we've had a lot of developments in our sequencing portfolio, but I just wanted to make it clear that Illumina is still investing heavily in the evolution of its microarray technology as well. So this ranges from workflow improvements to new content of both genotyping and our methylation array portfolio. Our goal is to ensure we have the most up-to-date content to empower more diversity in genomics and to capture the added value of multi-omics for any study. Next slide, please. In terms of our multi-ethnic genotyping portfolio, Illumina has had a long history developing such arrays for genotyping, which started with the launch of the multi-ethnic global array back in 2014. This was an array co-developed with the Kappa and Page consortia, who shared a common focus to increase the diversity of studied samples in genomics. This array, with 1.7 million markers, contains double the variation of the African's sequence from the 1000 Genomes Project, and was designed to substantially improve fine mapping and functional discovery by increasing the variant coverage across multiple ethnicities. Following on from the multi-ethnic global array, we launched the global screening array, which was designed to empower large-scale screening studies due to its lower price point and the most up-to-date clinical research content at the time. Last year, in response to the needs of the All of Us project, we combined the multi-ethnic coverage of the MEG array with the up-to-date clinical research content of the GSA to create the Global Diversity Array. Next slide. The Global Diversity Array was built for discovery and development of polygenic risk scores in multi-ethnic and admixed populations, but it also includes a large amount of clinical research and multi-ethnic exome coverage to identify high impact markers as well. It has a dense GWAS backbone with about 1.3 million markers, and it's also possible to add up to 175,000 SNPs of custom content, and this would include the psych array content if desired. Next slide. Moving away from genotyping and to our methylation portfolio, it's been quite some time since we launched the methylation epic array. This was back in 2015. And since then, we've seen a need to develop and expand our portfolio to cater not only for human studies, but also non-human studies as well. So at the end of this year, COVID permitting, we plan to launch a new human methylation array, which is focused for blood samples, which are the most dominant sample type for epigenome-wide association studies. This blood EWAS array will enable research such as population screening for known trait associations. It will facilitate epigenetic aging studies and also enable better white blood cell deconvolution, among other things. The array will target approximately 350,000 markers or beads and can therefore fit on our 24 sample bead chip format. So this means samples can be processed quickly and efficiently, so lower cost. For larger studies, it's also possible to customize the array with up to 62,000 additional beads. Next slide. In addition to our new human EWAS array, we will finally answer the prayers of many a mouse researcher and launch a mouse methylation array. This array is scheduled to launch in Q3 of this year, again, COVID permitting. It'll be about 316,000 bead types and will address many mouse applications, including epigenome-wide association studies, the dissection of patient-derived xenograft models, and of course, the analysis of mouse models of disease. It's also possible to add additional custom content to this array, and this is in the order of up to 10,000 beads. Next slide. So it's all good and well that we're bringing out these new arrays, but especially in methylation, but we understand that methylation studies have some other limitations, and therefore we've also been trying to break down some of the barriers to efficient methylation studies by working on new ways of managing the highly inefficient bisulfite conversion step. So we're working towards launching a tech note in Q3, which demonstrates a method for automating and reducing the time of the bisulfite conversion from 2 days to just 2 hours. So we see this as really critical for large EWAS studies where you don't want to be spending all your time and money converting your DNA. In addition to this, we're aware of new methods that can avoid bisulfite conversion altogether. So we're also looking into some of these methods in the hope that we can at least get one of them compatible with our methylation arrays so customers aren't constrained by limited sample. Next slide. And finally, we wanted to make you aware of our activities in the support of the COVID-19 crisis. We've partnered with many organizations around the world to ensure a rapid response to this crisis. Our sequences were used to sequence the first strain in Wuhan, and since then there have been many other positive collaborations to fight this threat. To further support our customers engaged in this fight, we're offering match funding based on a 50% discount up to $100,000 in value to any COVID-19 related activities. This is a short-term offer. It ends at the end of June, and all orders must be delivered for the project by the end of 2020. If you or anyone you know might be interested in this offer, then please reach out to your local salesperson, or you can reach out to Cora and I in the breakout room immediately after this presentation. So on that note, Cora and I would like to thank you for your attention today. And if you have any questions, we'll be happy to take them.
Okay, thank you both very much.